The immune system is broken into two parts, the innate immune system and the adaptive immune system. The innate immune system includes the skin that acts as a physical barrier, sweat that contains enzymes for pathogen breakdown, and the acidic environment of our stomach. The adaptive immune system is the main focus of our video, and that is split into two parts, cell-mediated immunity and humoral immunity. Humoral immunity is focused on special cells called B cells, which originate and mature in the bone marrow. B cells, once exposed to antigens, begin differentiating into two different types of B cells. One of these cells is called the effector B cell, which is responsible for the mass production of antibodies, proteins that are used to tag pathogens for destruction. Memory B cells are activated upon second exposure to release antibodies. The cell-mediated response is focused around special cells called T cells, which are lymphocytes that are formed in the bone marrow and develop in the thymus. Once exposed to antigens, the T cells mature and are able to commit to their functions. Cytotoxic T cells, or CD8+ cells, are responsible for killing infected cells. Once they interact with any antigen that has been presented on the surface of cells, they begin producing toxic chemicals that are injected into the infected cells, killing them via creating pores leading to lysis. Helper T cells act as the coordinating agents of the immune system. When they interact with antigens on the surface of cells, they begin producing cytokines. Cytokines are protein molecules that are used to communicate with other parts of the immune system. They draw in the CD8+ and B cells to the infected cell area in order to get the immune system to work. Helper T cells fall under the category of type 2 immunity, which are responsible for the production of multiple kinds of interleukin.
Now we will move on to one of the most important proteins that is involved in immune response, which are interleukins and cytokines. Interleukins are groups of cytokines that are expressed by white blood cells that mediate communication between cells. They are important in stimulating immune response, such as inflammation. That definition did not really help, so we will first define what cytokines are. Cytokines are small proteins that play an important role in cell signaling. They regulate growth, maturation, and responsiveness of certain cell populations. Cytokines modulate the balance between humoral and cell-based immune responses. If we say that the cell is helper T cell, the stimulus such as infection or antigen causes the cell to produce cytokine. When cytokines are bound to the receptor of a different cell, it sends signal to the cell that causes the cell to replicate and produce even more cytokines, resulting in certain biological effects like immune response. Some of these cytokines also activate cytotoxic T cells, and these cells are the ones that are actively killing the damaged and infected cells. There are many different types of interleukin, and they are designated with abbreviation IL followed by a number. 15 different types of interleukins are known currently, and they are ordered numerically from 1 to 15. For the sake of the Catalyst paper, we'll focus on IL-33. IL-33 is a member of IL-1 family of cytokines, and similar to interleukin-1, interleukin-33 increases the production of T helper 2 associated cytokines. Interleukin-33 is expressed by endothelial and epithelial cells, and high amounts are produced in those barriers. Because interleukin-33 is in the barrier of the tissue, it is the first immune mediator and acts as an alarm for cytokines. When cells are infected by diseases, interleukin-33 is secreted to alarm immune cells to produce cytokines. This is a basic schematic for interleukin-33, and this is the surface of a skin. When there is an infection or irritation by various factors like radiation, virus, and toxins, interleukin-33 is produced and travels to a receptor of a cell. This triggers the cell to produce more cytokines and replicate itself that leads to inflammatory response. The main concept of the paper is focused on the effect of a protein called HpARI on interleukin-33. HpARI protein is produced by mouse parasite called HES. That protein binds directly to active murine or human interleukin-33 and nuclear DNA to replicate proteins with interleukin-33 repressive effect, causing miscommunication or abnormal behavior in the immune system.
Another concept that our paper relies on is oxidation and reduction. From a chemical perspective, we understand oxidation as the loss of electrons and reduction as the gain of electrons. In the biological world, hydrogen can be seen as the electron equivalent in redox reactions. Oftentimes, hydrogen will be bound to electronegative atoms like oxygen, nitrogen, or sulfur. This has led biologists to define oxidation as the loss of hydrogen and reduction as the gain of hydrogen. To conclude our video, I'd like to tie everything together and back to our paper. Under normal circumstances, a reduced form of IL-33 interacts with its target ST2 to further the proliferation of other helper T2 cells. In our paper, it interacts with the protein HpARI, leading to its suppression, and as a result, the suppression of type 2 immunity.